So very good morning, sir.
Morning. Very good morning.
Good morning, sir.
Morning, sir. Morning, sir. Ma'am, you can start, ma'am. Sure.
Good morning to one and all gathered here. Welcome to virtual webinar on emerging technologies on coral reef conservation, organized by the Department of Biotechnology, DG Vaishnav College. Let us give a formal beginning with a welcome note. I now invite Divya Gomati of 2nd BSc Biotechnology to deliver the welcome address. Over to you, Divya.
Seeking the blessings from the Lord Almighty, I take immense pleasure in welcoming you all intellects for this virtual webinar. To give it a grand beginning, on behalf of the Department of Biotechnology, DG Vaishnav College, I express my warm wishes and welcome to Dr. Gerard Wilson, Assistant Professor, Department of Marine and Coastal Studies, Madurai Kamaraja University, for his interest and concern to be amidst us and to give insights on emerging technologies on coral reef conservation. I, I extend my warm welcome to all the delegates, professors, and graduates from all over India to be a part of this virtual webinar, and I am deeply overwhelmed by your keen interest in attending this webinar. DG Vaishnav College is a linguistic minority institution established in the year 1964 with the sole purpose of imparting knowledge and value-based education. Department of Biotechnology, established in the year 2000, focuses on developing young minds to be a competent biotechnologist with a profound expertise and innovative ideas to generate sustainable society in harmony with life and technology. Coral reefs, the rainforest of the sea, are some of the most biodiverse and productive ecosystem on Earth. They occupy less than 1% of ocean floor, yet are home to more than half of all marine species. Coral reefs provide food and resources for more than 500 million people in over 100 countries and territories. But tragically, coral reefs are in crisis. So, the methods and technologies to improvise and conserve the coral reefs has become sensational topic among the marine biologists in order to conserve the biodiversity of marine ecosystem. Conservation of Conservation is a state of harmony between men, land, and water. With this significant quote, I would like to invite Dr. Amali to introduce the speaker of this day. Over to you, ma'am.
Thank you, Divya. I'm extremely delighted to introduce our distinguished speaker for the day. Dr. J. Gerald Wilson is a renowned marine environmental expert. He completed his doctoral research in environmental sciences at Madurai Kamarajar University. As a certified scuba diver, he started his explorations in the underwater coral reef environment of Park Bay, Rameshwaram, and documented the benthic ecology for the first time. He has been awarded with various research fellowships from the Department of Ocean Development and ISRO Space Applications Center of Government of India. During his research pursuit, he has explored underwaters by diving at almost all Indian reefs, including Gulf of Mannar, Andaman and Nicobar, and Lakshadweep Islands. His career profile includes his profession as a scientist in the Ministry of Presidential Affairs, Abu Dhabi, during which he worked to restore the coral reef ecosystem of the UAE waters. He then moved to Saudi Arabia to join as assistant professor in the Marine Science Department at the King Abdul Aziz University of Jeddah. During this tenure, he explored the Red Sea water of Saudi Arabia and monitored the health status of the coral reefs. He had successfully transplanted corals in various non-coral environments. With 9 years of overseas experience, he is currently working as a guest faculty in the Department of Marine and Coastal Studies at Madurai Kamaraj University. To his credit, he has handled 3 major research projects and 2 minor research projects in Saudi Arabia, published more than 15 international research articles, and authored chapters in 4 books. He has presented research papers in many international conferences in India as well as abroad. He had been a part of the research cruise team expeditions to Gulf of Mannar. He has received the Young Scientist Award in the year 2017 for his innovative research in marine science, and recently he has been honored with the most prestigious Abdul Kalam Award for his outstanding contributions in environmental social activities. With this brief info, I'm extremely pleased to have you amongst us, sir, to deliver the lecture, and handing over the session to you, sir. Over to you, sir.
Thank you so much, ma'am. It's my honor to be a part of this webinar of the prestigious TG Vaishnav College. And I'm not basically I'm not a biotechnologist, but I'm a marine biologist. But I want to correlate how marine environment can be utilized with biotechnology, and what are the significance and the importance of corals, how we can conserve the corals, with some interactive slides presentation. And I welcome all you to the third word, that is underwater world. Shall I start my presentation? So today, this webinar is based on the emerging technologies on coral reef conservation. So most of you people already know what is coral reef and what is coral. There are many channels nowadays focusing on the environmental concern about coral reef conservation. So marine biotechnology, there is a small contribution from marine side to biotechnology. That is, without marine, biotechnology is incomplete, because most of the components and the particles are extracted from marine invertebrates and marine organisms, like collagen, which is helps in the improves our wound healing activities, and the various enzymes and proteins of marine life. They have a great importance in the biotechnological studies, especially in PCR. We are using some enzymes, thermophilic enzymes. These enzymes are extracted from the hydrothermal vents of extreme bacteria, which lives in the very extreme hot condition, so that the high temperature of the PCR can be possible nowadays. And similarly, cyclophiles, they can capable of dwelling in a low temperature, very low temperature. These kind of things are the gift from the marine to the biotechnology field. And there are some means the major contribution is from the coral reef environment to the biotechnological studies, especially in the form of drugs. Similarly, like anti-inflammatory compounds and anti-lymphogenic components, anti-proliferative compounds, then cytotoxic, and also antimicrobial, antivirus, and antipolyp compounds are extracted from the living organisms present in the coral reef environment, like corals, gorgonians, and others. So these are some of the extracts derived from the coral reef environment, and these are all the species which are contributed this kind of components from the marine environment. So the coral reefs, now we are going to brief about what is coral reef. So coral reefs are looks like a plant. See this picture, they are looks like a plant, but they are very bright in color. But they are animals because they belongs to the family and belongs to marine invertebrate of the phylum Cnidarium. Cnidarium means the animals which are having the stinging. So each coral is made up of tiny animals called polyps. So I'll go through one by one, so it will be have some good idea about what is coral and what is coral reef. So coral is a collection of small tiny animals called polyps. So like our honeycomb, there are many honeybees are gathering and constructing the honeycomb, right? Similarly, many polyps are gathering and constructing their own house. That is the exoskeleton. By the secretion of the calcium carbonate, they are building the core. So these corals lives in the compounds of colonies. So these colonies forms the reef region. So the major contributor of the coral reef is the coral. So many polyps are constructing a single coral, and many coral colonies are forming the coral reef environment. So, as in the welcome address, ma'am rightly pointed out that coral reefs are the tropical rainforest of the ocean because they are giving more and more benefits to the mankind. So, approximately 25% of all marine life can be found in the coral reef environment. That much of it vast and uh, very important because there are many crevices. You can see there are many fishes are swimming around here. So there are many crevices present in the coral reef, so that it acts as a feeding and a breeding ground for the other organisms. So that these organisms always prefer the coral reef environment, so that to pray that organisms, fishes will come. So to feed the small fishes, big fishes will come. So the environment will be very productive. So, but the corals are not present in all the seas. They are restricted to only the tropical seas. And in our India, we have a coastline of around 8,000 kilometers, among which we have only 2,705 kilometers square of coral reef. So the major coral reef environment of India consists of Gulf of Mannar and Palk Bay, which is present in Tamil Nadu near Rameswaram, and the Gulf of Kutch, Lakshadweep, and Andaman and Nicobar Islands. In this map, you can see this. So in India, we have only fringing and atoll kind of reefs. So what is fringing and what is atoll, we will see here. So there are three types in the reef. First one is called fringing reef. In fringing reef, the reef is present just encircled in the island. So barrier reef, there will be a water body in between the reef and the island. This water body is called lagoon. Whereas in atoll, only the outer ring of corals will be there without the presence of island. So the island will be absent. So this area now is called atoll. So this is fringing, and now this is the barrier, and this is atoll. Okay. So there are three kinds of types of reefs are there. So in India we have fringing and atolls, and the barrier reef is not present in India. So Australian Great Barrier Reef is the best example of this type of barrier reef. And as I said earlier, coral polyp is the fundamental unit of the coral. So this is the structure of the polyp. So they are just surrounding with the ring of tentacles. So these finger-like projections are called as tentacles. So they are usually multiple in six number. If you count this one, it's around 12 tentacles are there. So they are multiple in six in number in Scleractinian coral, whereas in octocorals they are 18, multiples of 18 number. They are sedentary, so they cannot move from one place to another. So they are fixed with the one place, so that they always looking for the prey. So they prefer in the current means strong wave action area, so that the prey will come by the force of water current. So they use the nematocyst present in the cell, that is the stinging cell, and it attacks the prey, sting the prey, and paralyze and capture the prey. So that they consider the animal and they put it in the phylum Nidarea. So this is about coral polyp. In each coral polyp, you can see the beautiful symbiosis of one algae called zooxanthellae. So in this picture, this brownish area, no, these are all the zooxanthellae. So this polyp gives the beautiful color to the coral. So without this zooxanthellae, the corals cannot get color. They are simply white in nature because their exoskeleton is consisted of calcium carbonate. So that this algae gives supplementary food and also bright color to the corals. So because this is algae due to photosynthesis, it gives color. On the other hand, it gets shelter from the polyp. So this is about the coral association. And there are two types of reproduction in the coral. First one is called sexual reproduction. In this sexual reproduction, there are two methods. One is the broadcasting type, and another one is brooding type. Brooding means the younger one, that is the planula larvae, is developed inside the coral, and it means it emerged out. But in the broadcasting means the sperms and eggs are just broadcasted out. The fertilization is taken place in the outside water. After fertilization, the planula larvae form, and the planula larvae is just floating in the water column and find a good surface, that means the hard surface. If its preferred surface is available, it settles and by secreting calcium carbonate, and it started growing by multiplication. So this is about the sexual reproduction, and we will see this video. One second, I'll share the video. So this is about the reproduction, sexual reproduction of the coral reef. So due to the full moon season, they have a synchronous spawn. So the entire environment of that region, they are having the mass spawning. It will happen due to the during the full moon season. So these eggs and the sperms are fertilized outside. So they forms baby coral, that's a planula larvae. So this is the planula larvae. It finds a suitable substratum here, and it settles down. After that, it's divided into two, three, four, eight, some. Then it forms a new colony over there. So this is about the life cycle, means the coral reproduction. So apart from this sexual reproduction, there is another kind of reproduction is there, that is called asexual reproduction. Here, the coral, that single polyp, no, This corallate is themselves divided into two. That is called the budding. So in the mother coral is getting divided into two daughter cells. This is called intra-tentacular budding. This type of reproduction. And whereas in another time here you can see a small coral, baby coral is formed in outside of the mother one. This is called extra-tentacular budding. So these two types of asexual reproduction is there. Apart from this, fragmentation is also there. So fragmentation means the colony is broken off from the small branches broken off from the larger colony. So these broken branches can be grown into a new coral colony. So this is called fragmentation. So sexual and the asexual both method both type of reproduction is present in the coral reef. So now we are going to see what are all the threats present to the coral reef. The first one is bleaching. Bleaching is caused mainly by the temperature anomaly. So the increase in temperature and decrease in temperature will result in the bleaching. Bleaching means the expulsion of the zooxanthal layer. So in this picture you can see the color is getting dull now. So here it become whitish now. So this is called bleaching. The entire zooxanthal layer will leave the coral when the temperature is not acceptable, because these corals are very sensitive. They are very sensitive to temperature, sedimentation, salinity variation, all things. They are very sensitive, so that they are restricted to some places. As I said earlier, these corals are present only in the tropical seas. But even though in tropical seas, they prefer only one place, one particular preferred place to go. That's why our Tamil Nadu is different. Okay, we have in mainland we have the largest part of coral reef in India. So here you can see this is the coral live coral with the brownish color. The same coral is bleached out due to the bleaching because of the temperature variation. So this will happen in every year. Sometimes in 1992 it happened, mass bleaching. In 1998 happened, 2000 happened. 2010 happened. So we are always monitoring the health status of the coral. So to conserve and predict the percentage and biophysical status of the coral. So this is the major natural impacts to the coral. So why the bleaching is happening? This is due to the global warming. So global warming because of the pollution. Ultimately, it comes to our mistake. So second one is sedimentation. Sedimentation means due to the cyclone and the storm times, the entire water means the seawater will be very turbid. Turbid means it has a very the sedimentation will be very high. So during calm season, you can see the very clear picture. So this is called the sediment trap. These traps are placed to measure how much quantity of sedimentation occurs by monthly. So due to during rough season, we cannot see that the area clearly, no. So this is the just the example for how much the sedimentation occurs in the coral environment due to the monsoon season. So once the sedimentation is turbulent, the water column is turbulent, the upward sediment will finally settle on the coral. So here, one soft coral is completely smothered by the silt, isn't it? So by what happens, this silt and sediment finally reaches the mouth of the coral and it blocks the mouth, so that the polyps cannot be extended. So without extension of polyps, they cannot take the food. So finally, the coral will die. In this picture, you can see the left-hand side is the healthy coral, whereas the right-hand side the completely darkened corals. It's already covered with the other bacteria and the marine algae. So this is a partial death. So finally, what happens? This algae and the other microbes will attack other healthy coral also. Then they can't hold the place. So this is sedimentation and developmental project. Nowadays, it's a very commonly we can see in ECR and other areas. many projects are developed in the seashore area. So the constructions that happened just on the seashore. After tsunami only in India, they stopped constructing. They banned constructing buildings in near shore area. But in some cases, they are just ignoring, especially huge industries are mainly placed on the seashore. And what they simply dump their uh, waste and the drainage sewage water in the marine environment. So finally here, the sewage water from a city is pumped inside the marine environment, which is a coral reef environment without any proper treatment. So these kind of things will completely damaging the coral. So another kind of impact by the human is the trap fishing. So this is the left picture shows the trap. So these kind of traps are placed in the coral reef, the gaps between the corals. They put some bait inside so that big fishes will come and they got entangled. So sometimes the fishermen don't know where they put the traps, and they simply abandon the traps. So these traps will cover the corals and they damage the coral. And sometimes these traps are made up of very strong iron, cast iron. So these iron rods are damaging the corals because corals are very fragile in nature. So their growth rate is very meager. So for 0.5 mm to 3 mm, it takes around a year to grow some corals. So, but we can easily destroy because they are made up of calcium carbonate, no, like our chalkfish. It is very brittle in nature. So these kind of traps are damaging the corals, but it takes years to grow back. And this one is a very worst practice of illegal fishing, blast fishing. So this fisherman is just putting a dynamite inside the coral reef environment. So the dynamite will blast inside water, the underwater, and all the fishes in the vicinity will become die and they float. So they will simply go and collect this one. This will also damaging the corals. And this is oil spill. So in my first slide, I said without the marine environment, there is a no biotechnology. No? So here the things is changing. Without the biotechnology, we cannot overcome the oil spill. So there are many new technologies are emerged from biotechnology like bioremediation. So these kind of things are nowadays used to digest the oil spills. They produce super bacteria to sorry to digest the oil spills. So the ship grounds and the spill of oil will finally enters into the coral reef environment and the entire animals living organisms present in the area become dead. So these are all threats are available to the coral reef. So if the coral reef is not there, we cannot get much drugs and other marine natural products from the coral reef. So we have to conserve the coral reef. So how we can conserve the coral? Here comes the fragmentation. So in the first reproduction, I said no. In the asexual reproduction, there are two methods: budding and fragmentation. So the fragmented coral can grow into a new colony. So this idea was developed into the transplantation technique. So the small colony is taken off from the branch and it was planted in the another area. So here I show you video how they are collecting the um, colony from the coral. So small branch is collected from the coral colony. Actually, it's a very small colony, so we collected the entire coral. So this is Acropora species, very fragile in nature, and they have a best growth rate. So we prefer to transplant this kind of corals which having good growth rate. So this is called nubbing. So we are taking this nubbing and we are place them in the table. Like so, these kind of iron tables are made, and these nubbings are fixed with some underwater glue or underwater cement, and they acclimatize in the same area where they are collected. This is called acclimatization for some period. So, this is one type of nursery. This is another type of nursery. This is called the tree nursery. So, in this thing, this one is better than this because here sedimentation will sometimes kill the baby corals, that is the nursery corals. Whereas here, sedimentation will not have that much of that because they are hanging in the midwater. So this is otherwise called as midwater nursery. So after 6 to 12 months, they become this much of big colony. So after that, these colonies or small nubbins are transplanted into the area where they don't have much diversity of corals or they don't where the new area which we can establish a new nursery. So this kind of things is called transplantation. Another thing technology is called translocation. So this translocation means we have to instead of taking a small branch from a colony, we are just moving the entire coral colony from one place to another. This is mainly this type of translocation is useful for the places where some oil refineries in Gulf countries now there are many oil resources are available offshore area. So if they find a new oil resources in the offshore, immediately they want to put some constructions and the pipes to the shore area. So definitely that will damage the environment. So that time what the people do, we remove the entire coral reef in that area. So this kind of here the one diver is selling and from the base and removing the entire exact the coral colony. So these coral colonies are just to put it in the water basket and then transported to the nearby area where the um, impacts on the damages and the anthropogenic activities are not very less. So that area we have to place the coral by using the underwater cement. So here, this one is the new environment where the collected translocated corals are pasted. So after some time, they will grow fully. These are all the very basic methods of transplanting. And one novel method recently, actually, it developed in 1980s, and now it's getting good percentage that is advanced to one. This is called electrolysis of seawater. So the thing is, while constructing the exoskeleton of calcium carbonate, these corals are taking the calcium present in the seawater and the atmospheric oxygen, that is carbon dioxide. So calcium carbonate will be formed. So they want actually there is a process of lysing the water and take the calcium from the uh, dissolved water condition. So this electrolysis is doing the thing to enhance to enhance the calcification, the electrolysis process. So the calcium will be accreted very drastically because the corals cannot that much. They have that much potential to extract calcium. So they are giving this kind of boost to increase the mineral accretion. So this is nowadays very famous. In many countries, they are using underwater museums. So they put this kind of different shapes and statues underwater, and they fixed the coral patches here and there, and they put electrical, very battery electrical current with the anode and the cathode. So this will electrolyte the calcium present in the water, and it enhances the growth. So in the, within 6 months of time, we will get a good result in the this kind of things. Okay, so these are all the technologies uh, we can use to uh, develop or sustain or conserve the coral reef. So from he's Dr. Jacques Coste. He's called as the father of scuba diving. So his quote is, if our grandchildren never have the opportunity to see living coral, it will be the everlasting shame of our age. So, so means so we have to conserve the coral reef environment, not only for our biotechnological purposes, but also for our grandchildren, to the betterment of our life. So I stop here. So if you have any queries or questions, you can raise now. Thank you.
Thank you, Dr. Gerald Wilson. That was an eye-opening session on the significance and the need for the conservation of coral reefs in marine ecosystem. Now we'll move on to the interactive session. The participants can post your questions in the chat box, which will be answered by the speaker. Sir, here is a question from Harsha of Biotechnology Department from DG Vaishnav College. Does the excreta from any of the fishes that reside in the corals, such as parrotfishes, play any role in bleaching events?
No, actually, this excreta are taken by, there are many detritus feeders are present in that coral reef environment. So whenever the waste, may even dead fish or excreta from other organisms, they are eaten by other associated animals. So this is not a major issue. But the parrot is, some parrot fishes are feeding on the corals. So they used to click the coral colony, and they eat polish, so that it may damage that colony. But that excreta will not.
Fine, thank you, sir. Next, we have a question from Chandru Patel. He's asking whether the coral reef is capable of growing only in seawater, or is there any species available in fresh water?
Actually, corals are only restricted to the seawater. So they need the optimum salinity of 35 parts per thousand. So that's impossible for culturing or growing corals in the freshwater.
Fine, thank you, sir. And here is one more question regarding the oil spills. So basically, if you see oil spills, they will be deposited on the surface of the seawater. How does it affect the environment, the coral reefs, which is just beneath the, underwaters, which is present in the underwaters?
Yeah, actually, the corals have that associated animal, no, plant called zooxanthellae. These zooxanthellae needs sunlight to photosynthesis. So if that oil spill is covered that entire surface area, the sunlight penetration will be stopped. So without the supplementary food, the coral is starving to death. This is one reason. And next reason is for constructing the exoskeleton, it needs the carbon dioxide from the atmosphere. So due to wave action and the water movement, the air and the water mixture is different. So the air water is mixing with the sea water. so that the corals will take that carbon dioxide and with the dissolved calcium, they make their exoskeleton. So without that one, it will be very in danger. So that these things are will damage corals during the oil spill.
Fine, thank you, sir. And here is one more question from Dr. Ashwini. Can we find any halophilic archaebacteria in coral reefs?
Halophilic bacteria. Actually, I'm not, as I said earlier, no, I'm not a microbiologist or biotechnologist. I'm a coral reef ecologist. So there are many bacteria and things are means the halophilic bacteria are present in the coral reef environment, but I don't have that much of details about that one.
Yes, sir. Thank you. And here we have one more question. Can you just describe the impact of bleaching effects on coral reef, both positive and negative?
Actually, bleaching it contains only the negative impact only. There is no positive impact. So bleaching, similarly, actually, the previous question about oil spill also related with this one. So bleaching means the expulsion of zooxanthellae from the coral. So this zooxanthellae that is associated with the algae, it gives the supplementary food to the coral because corals are sedentary. They cannot move from one place to another to hunt their prey. So they have to wait until the prey comes to the coral to catch and paralyze that. So mainly they are depending on the supplementary food produced by the zooxanthellae. Zooxanthellae is not there. Immediately what happens? The coral will stop. So only the water current which brings the food can be replaced by the coral. That whenever the salinity variation, temperature variation happen, immediately the zooxanthellae will leave the coral. If the environment is normal within some time, maybe 1 week or 2 weeks, then they will come back and sit on the same coral. Otherwise, they will. So if the coral is dry, immediately that coral is occupied by the marine algae and it will overcome and the entire reef will be damaged. If the coral is dead, immediately it starts deteriorating. So the coral will become rubble, that broken corals, and finally it reaches the surf means sea floor as a calcium deposition.
Fine, thank you, sir. Here we have a question from Dr. Gayatri. She has complimented on your presentation, saying that it was a nice presentation, a nice session. Can you please name the dominant coral species in East Coast?
Usually, in the means very like 90s and 80s, from 70s onwards, Acropora are dominating in the southeast environment. But after the bleaching event of 92 and 98, the Acropora, that is the branching coral form, they look like a tree, and they usually called as a stag form because they look like the horn of the deer. So these corals are drastically damaged and their population is now very limited. So only the massive ones, and according to my knowledge, the Favia and Corycus, these corals are dominating in the southeast.
Fine, thank you, sir. There's a compliment from Dr. Marimuthu stating it as an excellent presentation and he has congratulated you. And there is one more question, sir, from Harsha. There was a study that showed that microfragmentation of coral reefs can help in growing them rapidly, like how the skin of mammals grow rapidly when it is cut. How effective can this technique be in growing them?
So this is a very interesting question. So micro fragmentation. Actually, during the transplantation, scientists will take the major portion of the branch, no, like a nubbin, like a one finger kind of around 3 mm to 5 mm size they will take. So this will also destroy the environment because of the wound and the scar. the mother coral also having some issues. So to overcome this problem, this micro fragmentation is invented in Japan. So this micro fragmentation means they have to take a small portion, like our microsome, we are taking a small portion. No? Similarly, they will take a small portion and they culture into a new coral reef or new coral colony, the laboratory purpose. But as I said, corals are very, very sensitive to environmental anomaly. These corals are they are culturing this kind of micro fragmentation is a very a kind of a Herculean task. So we need a flow through system, a running water system, and we have to make no, like nurturing a baby. Very difficult to take that microfracture. But many scientists are success this one. If they have in onshore lab, they have the supply of good seawater, they can go for this kind of microfragmentation. And for reef aquarium also, many aquarists are doing this kind of thing. They culture by microfragmentation and they are selling the artificially means in nursery culture course.
Fine, thank you, sir. Here's one more question from Vardhana Janakiraman. She has asked whether this COVID lockdown had got any positive impact on coral reefs.
Yeah, definitely it has some positive effect on the corals because there are many kind of other pollution which I am not touched in my presentation. It is called that cosmetic pollution. Usually the recreational people know these people are always using the sunscreen lotion while they go to beaches and snorkeling or diving. These sunscreen lotions have some kind of impact on the marine environment. Particularly, there are many studies shown that they have a very worst impact on the corals. So, due to this lockdown period, this pollution is completely stopped. And next thing is the vehicle movement is already stopped, reduced. So that the atmospheric pollution, the air pollution is already reduced. So that it has the indirect effect on the coral. So the global warming or the temperature nowadays we are getting periodical monsoon. Now in India, the monsoons are not that much predictable. No, now we are getting the monsoon in the entire means the exact season we are getting the monsoon. Similarly, the COVID lockdown is having a good effect on the corals. On the other hand, there are some other impacts also there because the use of masks and PPE kits are simply thrown in the sea. The people are thinking ocean as a huge dustbin. After usage, all the waste materials are simply dumped in the sea. So you see Goa River due to during the tsunami, it the entire fish in the Goa River. So finally, all this plastic material and mass entering into the sea. So this will cover the coral and suffocate the coral too. So there are both the positive and the negative impacts are there.
Fine, thank you, sir. Now we'll conclude, get to the conclusion of the session. And there are so many, before that, there are so many compliments and appreciations regarding your presentation, sir. And thank you for so much for your availability. And you have made this program a great success. A good start, of course, will have a pleasant ending. I would like to now invite University of Second BSc Biotechnology to conclude this webinar with a word of thanks.
On behalf of the Department of Biotechnology, DG Vaishnav College, I deem it a pleasure to propose the vote of thanks to all of you for being part of this virtual webinar. Foremost, I would express my sincere gratitude to the Lord Almighty for making this session as a remarkable platform of knowledge sharing with grand success. I express my sincere thanks to our college management for the constant support. In a special way, I would like to thank and acknowledge our secretary, Sri Ashok Kumar Mundra, and our principal, Dr. S. Santosh Babu, for their motivation and constant support. I'm extremely thankful to our distinguished speaker, Dr. Gerald Wilson, for accepting our invitation and enlightening us with his expertise on reefs and their significance and the methods to conserve them in order to maintain a sustainable marine ecosystem. I extend my sincere gratitude to Dr. Balakumaran, Head in Charge, Department of Biotechnology, for his endless support, encouragement, and gratitude in organizing this event. My sincere thanks and due to the organizing secretaries, Ms. G. Shobha and Dr. Amali, for this meticulous planning and execution of this event for minute details. Furthermore, I would like to extend my sincere thanks to all the delegates, professors, and graduates for your support and interest in attending this session to enhance your intellectual horizon. In pace with the famous quote, alone we can do little, but together we can build a castle of knowledge. I'm thankful to all the faculty and students of Department of Biotechnology for this endless support and voluntary help. Once again, my sincere thanks to each and every one of you in making this event with an echoing success. Thank you, one and all.
Thank you.
Thank you, sir. Participants are requested to kindly fill the feedback form to get the e-certificates. The e-certificates will be dispatched within 2 weeks. Thank you.